at the ICLE, I presented 2 studies. One of them with the group of Mariette Melli at the Amsterdam University Medical Center. The group there actually showed that adding a secondary costimulatory receptor, so a chimeric costimulatory receptor, to a CAR T cell enhances the overall avidity, which results in better cytotoxicity, both in vitro and in vivo. Now, one of the exciting parts of that study is actually that when you take out the signaling domain of that costimulatory receptor, we actually see that only enhancing that cellular avidity by enhancing the valency at the cell-cell interface already increases cytotoxicity and actually the efficacy of the CAR T cells, which shows that cell avidity as a measure is really upstream to the functional outcome of these therapies, which usually is cell killing. The second studies that I've presented here was work that we have done in conjunction with the lab of Steve Rosenberg at NIH. They isolated their T cell clones that were specific for the G12D KRAS mutated peptide from a pool of patient-derived tumor-infiltrating lymphocytes after TIL treatment. And actually what they did prior to our coming to the lab, they followed the expansion of these T cells, of these clones, in the peripheral blood of the patient, which is indicative to how successful they are in battling the cancer. We measured the avidity of these three of these clones, and what we've actually shown there is that the affinity of the T cell receptor, the specific T cell receptor to the peptide MHC complex, does not predict the avidity of it, the avidity score, and does not predict the persistence of these T cells in the patient. So actually, the T cell clone that had the highest affinity did not persist at all in the patient, and the same T cell clone that had the highest affinity also showed lower avidity than another clone that had intermediate affinity to the peptide and did persist in the patient. Actually, with that, we showed that avidity by itself, cell avidity, is a better predictor of T cell function than the affinity of the single receptor to its ligand.